All right, well, good evening, everyone, and welcome to Nebraska Mycological Society Spring Mushroom Series Part 3, The Key to Mushrooms, Beyond the Basics by Rebecca Chandler. Rebecca Chandler is a butterfly conservation specialist for Nebraska Monarchs, certified ethnobotanist through the University of Alaska, Nebraska Master Naturalist, and lifelong horsewoman. For the past 10 years, she has worked in the field of conservation, local food systems, and outdoor education. Rebecca loves foraging for wild mushrooms and sharing her passion with others. So welcome, Becky, and thanks for joining us.
Hey, thanks, Derek. It's great to hear, and thank you all for tuning in. It's really been a long-term, dream and hope to have a mycology club here in Nebraska. So I'm really excited about this. This mushroom series, spring mushroom series, has been really fun to see how each of our talks has been building on each other, and I'm excited for Jared Adams' talk next week as well. I know that all these talks are being recorded, so for those of you who aren't tuned in right now, you can tune in later. So a little bit more about my how I got into mushrooms. I grew up in Western Nebraska, out in the country, so I was always kind of running about outside, and I can't really think of a definitive moment in which I realized I loved mushrooms, but I was always fascinated in just seeing them all around. I worked in Yellowstone and Olympic National Parks for a couple summers, so there's just a huge variety of different mushrooms, and that's when I started picking up my field guides and working through keys and trying to understand the different terminology in order to be able to identify different types of mushrooms, different species. So it's a huge world out there, and it's really exciting. Just I'm always learning more. So we're really lucky to have a super knowledgeable group in the Mycological Society right now. So I will, let's see, I have the power to move the next slide. So my talk is going to be pretty broad. I'm going to go over a lot of different morphological characteristics and terminology, and tips for foraging, for getting started. And one of the things I think is it's a small hurdle for people just getting started, and for myself included, is really sensitizing your observation skills to notice all the tiny little features that you can find in different mushrooms. And here I've got Hypholoma capnoides, or chicken of the woods, up top, and there's what I believe to be a scurfy twiglet on the right there, a Tuberia species. And sorry, the chicken of the woods is a Letoporus sulphureus, and the other one at the bottom left, I was thinking of Strophiaceae genus. Let's see here. So some of the tips that I have just for getting started is starting with one or two species at a time and trying to get to know them as well as you can. Looking for poisonous or lookalikes is also pretty important, just because you can rule out the ones that are toxic, and you definitely don't want to eat. So I would recommend starting with some of those. Also, just spending some time with the field guides before you go out in the field. A lot of them are just organized differently, and you don't always want to be flipping through different pages while you're there in the woods. You want to be enjoying yourself, and so I would take a look at those before you head out. And as just careful observation at combinations of the features. So, not just the cap color, not just the stalk length, or, take a look at how that mushroom, its entire life cycle, and also just all the combinations of spore print and spore color and gill spacing, and just kind of combine those all into one big picture. And sometimes it is helpful to do some drawings too. I like to draw plants and mushrooms when I'm out there observing. It kind of helps me to lock in those different characteristics. Another thing is you can cross-reference multiple field guides. Most field guides have some bias to them just because that specific mycologist is based in a geographical region, so they're more accustomed to the mushrooms in their region. So it's good to cross-reference and compare and contrast. Learning to make spore prints is also super helpful. After I jot down, or if I'm either sketching or writing down what I'm observing, I also will do a spore print right away because that can narrow it down a lot right away. And lastly, just, using your best judgment, because only you really know if you are confident in that identification. So don't always listen to, what other people say, or on Facebook, there's a lot of identification groups by pictures, and, it's good to ask for help and have those identifications through pictures, but really you want to be confident in it yourself. And the only way to do that is to do your own research. So let's see here. Another thing, if you're collecting mushrooms for identification, you'll want to record the surroundings, take it with you, segregate the different species into different containers so you don't get them confused later on. And I know you might think, while you're looking at them, you're like, I could never confuse those, but if you get enough in there, it can happen. And there's a lot of mushroom underneath the ground, so it's helpful to collect the whole mushroom if possible. And if you're doing that, try not to disturb the environment, the substrate too much while you're trying to do that. And again, spore prints are really helpful. Using keys, kind of the words versus pictures, it's important to use keys because you can't use pictures alone, and you can't really use just verbal descriptions alone either. You want to use that in conjunction with the keys. There's a lot of subtle differences in mushrooms that you can't really produce exactly in photos or writing. So that's why where keys come in. Also, a thing about spore prints is that they don't change in color as the mushroom matures, whereas a mushroom, when it's in an immature stage, can look totally different when it's in a more mature stage. One of the things, I'll be going through a lot of kind of technical terminology and morphology. Hopefully I won't bore you too much on that, but if you're a little bit of an etymology nerd like I am, I think you might find it really fascinating. And so it's also important to know some morphological groups and characteristics, understanding binomial nomenclature, the two-name scientific structure of Latin names, and when you feel confident enough that you've done all your research, you've cross-referenced field guides, gone through some of these identification tips that I'll be going through, then you might feel confident to eat a mushroom you forage. But again, I'm, only you know. So we'll go with that. So field guides, these are some of the ones I really like using. Mushrooms of the Midwest is by Michael Cuyo, and it's a really great Midwest-based field guide. The kind of tome of all I love the book Mushroom Demystified. It's just really well researched, super comprehensive. There's over 2,000 mushroom species listed in there. It's great for beginners and advanced mushroom hunters because it gets into some more technical details. He even talks about like measuring spores and different microscopic features for those who do have microscopes. And he's just really hilarious guy. He writes in a super quirky, entertaining way. He's also and this book is thick. It's like, I don't know how many pages, probably, 700 pages. So it's not something you'd really want to go schlepping around in the field with. But David Aurora also has a little field guide, all the rain promises and more. So it's more convenient sized for taking out into the field. There's a nice little ruler on the back side of the cover that you can use. And David's based out of California, so it might be a little bit more swayed to those West Coast species. So that's just something to take into account. And Gary Linkoff's The Audubon Society Guide to Mushrooms, great. It's pretty comprehensive. There's over 700 species there. It's got some full color photos. It can be a little bit harder to use just in the way it's organized. The pictures are separate from the keys, so you kind of have to flip back and forth a little bit. But once you're familiar with it, then it's not too difficult. I also really like the how to identify mushrooms to genus with macroscopic features. It's just really great illustrations. There aren't any full color photos, but really great drawings that I appreciate and I think are really helpful. He's got another book also with microscopic features, but it's a little bit harder to find. It's a little more expensive because it's not in print anymore. Same with this one. You might find it used. I think I found mine on the Abe Books or something like that. So, but it's a great, pretty small little book to have. I really like it. So here's an example. I'm gonna go over some just different types of keys. I think that a pictorial key is really helpful to start out with, just because it's super basic, and this is a pictorial key of major groups of fleshy fungi. It's in David Aurora's book Mushrooms Demystified. And so it just goes by characteristics and spore print color. So all, macro features, and it's super fun and easy to use if you're a visual learner. It might help you to identify a mushroom, at least to the genus, if not the species, but at least the genus. Another type is a tabular key. It's also pictorial and very much the same as a pictorial. The flowchart key is, this is for pink spored Agaricus. And I just think it's kind of interesting because it's a lot easier to backtrack using a flow chart like this. You know exactly where you came from. So if you need to backtrack, it's very easy. And that can be a lot more difficult when you get on to like cotymus keys that are longer. And then there's some really good online keys on online sources. Michael Cuyo's Mushroom Expert is an awesome source. He's got all kinds of really great photos and glossaries and keys. Messiah College Key is a good one as well. Great for, visual learners. And Ed's eKey it's a digital port that's public. And let's see here. And then the MyCokey is another downloadable and interactive one. That's really great. Okay. So yeah, using, dichotomous keys, it's not really complicated. It's actually a very simple kind of choose your own adventure type system. You just start with number one, and then you choose which one you find to match what you're looking at, and go on to number two, and so on and so on. You only have really two choices to choose from each time. So this is the key to the Agaricus genus, and it's a really large family of mushrooms. It's important to know that you don't have to keep going if you feel like you're unsure about one of the choices you made. You can always go back, and sometimes you do get stuck with keys, and it takes a lot of patience, and that's why it's super important to just have different keys available to you, different field guides. There's a few different reasons that a key might fail. It might not be really properly organized, the key that you're looking at. The mushroom that you found might be not in that book, or not even yet identified yet. So it's just good to have different resources. Another thing I want to just touch on a little bit is LBMs, little brown mushrooms. They're kind of most mycologists' worst nightmare, just because they're notorious for being very difficult to identify. The definition of an LBM is a gill-bearing mushroom with a cap no more than 5 centimeters or 2 inches in diameter, that has spores, gills, stipe, and cap that are all the same shade of brown. So this is a Gallerina autumnalis, or a deadly Gallerina. It's considered an LBM, and it's toxic, as its name says. And these aren't for novices, and really it's better to start with larger specimens that are easy to identify and have easy to recognize characteristics, than to start with something like this. Not that you can't, but it's best to kind of start with the low hanging fruit as you kind of get to know it and up your confidence with identification. Let's see here. Okay, so I'll talk a little bit more just about spore prints, because this will be a really important thing to know how to do when you're identifying mushrooms. So this little chart here is nice. I've heard so many people say that they just like to use white paper, or use foil or glass, or everyone has preference for how they can see color the best. Personally, I do like using just white paper for almost all of the spore prints I do. I find that the white paper, even if it's just slightly beige toned or slightly lavender, I can see it better. I can at least see the tone or the color a little bit better. But still, this is kind of helpful. And let's see, and I feel like the spore print is kind of one of the most helpful tool if you're trying to get mushrooms kind of narrowed down to a genus quickly. Here. So it's it's cool that you can take a spore print right after a light rain. That's generally when they're dropping more spores. That way, when you get home, you don't have to put the water onto the cap to get them to drop the spores. So you can use white paper for light colored spore prints. And if it's a super mature species that's dried out a little bit, or even very immature specimen that it might not produce a spore print. Same with polypores, it takes a really long time for them to produce a print, and they might not either. So you can also use air currents if you want to make some interesting patterns, kind of make it more artsy. This is a picture I took of Pleurotus that you can kind of see that lavender tone of the spores it released onto the tree. So I thought that was really cool that you don't really even have to wait till you get home. If you just look around, there might be some staining on the gills or staining around the tree that the spores already fell off onto. And I'm just going to touch a little bit on the chemical characteristics. There's two main chemical reagents that a lot of serious mushroom hunters like to use, and that's KOH and the, let's see here, Meltzer's reagent. I haven't used these. They're a little bit tougher to come across, but I know that Chance has done some KOH reagents, and so I'm just going to talk a little bit about it so you all know that this is a really good way to identify mushrooms. With the Meltzer's reagent, if it stains a bluish gray to bluish black, it's said to be an amyloid. And if the spores turn brown or reddish brown, it's a dextrinoid. And if you use a stronger solution of KOH, it can turn this specific mushroom, Hapalopilus nidulans, very purple. And I just think that's really beautiful color. It also, this mushroom makes a nice dye if you're doing any mycopigmenting. Okay, so before we get more into using dichotomous keys, I'll talk a little bit about the classification of the fungi kingdom. And so you start with kingdom and go to phylum, class, order, family, genus, and species. Agaric mushrooms are their spores on gills, and they're by far the most familiar and numerous and complex order of mushrooms. Originally, they were all grouped into one massive genus called Agaricus, but then this guy Elias Frias, in the 19th century, he divided Agaricus into smaller genera based on their easily ascertained features. In Mushrooms Demystified, David Aurora divides his into 14 families, divides the group Agaricus into 14 different families. And I'm not going to spend too much time on this because I know Rachel did talk about this in her talk. And so it's important to know just some of the major features and anatomy of a mushroom, again, so you can read through your economist keys and know what they're talking about. The pileus, also known as the cap. There's the hymenium, which is the spore surface. The annulus or cortina, reticulum, stipe or stalk, and base. And at the base is also sometimes there's a vulva, which is a cup-like structure that's the remnant of universal veil. The annulus or cortina, that's a remnant of a partial veil. So if you imagine the whole mushroom, if there's a universal veil, the whole mushroom looks like an egg. And then as the stalk grows taller, it tears. And so there'll be remnants of, sometimes on top, it looks like on Amanita species, you'll see those spots. And that's part of the leftover universal veil. And they also have a vulva at the bottom, which is that remnant as well, where it tore. So yeah, you can kind of break down mycological characteristics into these different ones. When you're taking notes, you could even like break this down into a checklist, print it off, and then either a checklist or kind of fill in the blank. That way you don't have to write it out every time. And that way you can quickly just fill in the blanks that you can observe while you're in the field. Hymenean types, we'll go over that. So here's different cap shapes: convex, bell-shaped, conical, knobbed, flat, and sunken. Let's see here. And then the cap surfaces, there's smooth, velvety, hairy or fibrous, ridged scales, flat scales, and patches. And I know this is a lot of information I'm going through really quickly, but all of this information is available in a lot of field guides and different online resources. So it's mostly important that you just know it's here and what to look for. So, so there's also the attachment of stalk: adnate, annexed, notched, succeeding, descending, and free. Here's some really good pictures from Michael Kuyo's site that are just the spacing of lamellae: distant, subdistant, close, and crowded. Different spore-bearing surfaces. You've got gills, smooth tubes or pores, the kind of blunt-like ridges. A lot of the Hericium species have those teeth-like structures, and then some puffballs have well, most of them have a gleba, so that's just that kind of solid surface when you cut them open. Here's some different margins of the pileus that you could take note of: smooth, crenate, striate, wavy, appendiculate, remose, or cracked. Here's the annular rings, pendant, flaring, sheathing, double cobwebby ring zone. Different stipes and stems as well look differently. There's equal club-shaped bulbous with cup or vulva, ring, and with rhizoids. So yeah, more information on features to observe and record. Using all that anatomy that you were just looking at, you can kind of start to measure different parts of the fruiting body, notice its stature, shape, mention what the nature of the spore-bearing surface is, if it's pores, gills, teeth, tubes, the color of the spores, And also you can note the color of the gills, cap, and stalk, although taking note that that might change, is likely to change as it matures. Arrangement of gills or pores, gill attachment to stalk, texture of cap, stalk, distinctive markings, and does the cap or stalk change color when bruised or handled? Does it exude a latex or a lot like a Lacteris species? Also, smell the mushroom and see if there's an odor to it. A lot of mushrooms have different odors to them. Cucumbers, watermelon, licorice, mealy. It's really fun to see all the different kinds of smells they have. And then there's the character of the pileus, character of gills, character of stalk, growth habit. Is it clustered, solitary? What's it growing on? Roots, rotting wood, live trees. Some grow on insects or pine cones. And again, spore color. So there's really a lot to observe. Okay, so now we're going to kind of switch gears over to the more microscopic characteristics. Now that I've gone through all of the more macro features, even if you don't have a microscope, it's kind of fun just to see what it looks like anyway. So first off, we've got the Ascomycota. Ascomycetes bear their sexual spores in a sac called an asci. And so there's cup fungi, flask fungi, truffle-like fungi, and morels also are Ascomycota. So, sometimes in those asci structures, they look kind of like a legume. There's usually 8 spores, but sometimes there's 6 or 4. And so if you have a microscope and you're looking through that, just knowing how many spores are in that sac structure, you can help identify it a lot more confidently. And so then there's also Basidiomycota, and they bear their sexual spores externally on these club-like structures called a basidium, and that's why they're often called club fungi. And so there's the hyphae with the basidium out of it, and then the spores on that basidium. And there's generally 4 spores. And let's see here. Some more micro morphological features that you can take a look at. This is an excerpt from the Mushroom Demystified. So yeah, again, it's just kind of meant to give you an idea of what they look like. You can go on to different websites and look up, pictures, type in arrangement of gill hyphae of specific mushrooms, and people might have submitted them. Mushroomobserver.com is another really great website to check out for those microscopic photos. So here you can see the arrangement of the hyphae as seen from a cross section. So there's interwoven, there's parallel, divergent, convergent. Another thing you could look at is the structure of the cap cuticle, scalar, and filamentous. And then also the cystidia. The cystidia are pretty relatively large cells that are found on the sporocarp of Basidiomycetes. And the spore sizes are measured in microns, which a micron is 1,000th of a millimeter. So in order to see some of these structures, you need at least 400 times microscope, but really you'd want like 1,000 at least. And so here's some different spore shapes that could also help you with identifying a mushroom species. I won't read through all of those, but as you can see, there's a lot of different spore shapes to look out for. Some really cool different shapes. Another micro feature you could check out is the spore surface. So I'm not going to go over too much about the mushrooms and their environment because I know Chance in his talk, he discussed a lot about this. So I will just kind of breeze over that. There's three main substrates that or ways that mushrooms interact with the environment: saprophytic, parasitic, and mycorrhizal. The saprophytic mushrooms is the kind of the mass majority of fungi, and they subsist on dead or decaying matter, so wood, humus, soil, grass, dung, and other debris. So these are the decomposers, and they release acids and enzymes to break down the dead tissue into smaller molecules that they can then absorb. So these types of mushrooms can create fairy rings. One of the largest mushrooms, the humongous fungus, is in Oregon, and that's this type of mushroom. There's also fairy rings that you can find in like Midwest prairies that are found to be like 600 years old. So, and other, so reishi, shiitake, oyster, turkey tail, dryad saddle, those are all saprophytic. There's also mycorrhizal. These fungi receive sugars from the plant in exchange for nutrients, water, protection against pathogens, and this is a symbiotic relationship. Oh, okay, not yet. Okay, so we'll go over some of the mushrooms, and you can kind of try to use your observation skills based on all that all that different anatomical information we just went over. So for this one, I'll kind of just describe it as you look at it. So the cap is bright yellow orange color. It's got decurrent gills, meaning the gills go all the way down the stalk a bit. The spores are pale. Can't really tell from the picture, but you kind of can. And it's got a tendency to grow in clusters. This is a cool mushroom in that it also has a bioluminescence. It can be blue-green color that's observed only in the really fresh specimen in low light conditions. I've never seen it before personally, but I've seen some pictures and it's really cool. I would love to see, I think they call it foxfire, but I would love to see that someday. So here's the gills. Like I said, they're decurrent and closely spaced together. So this is the Omphalotus olearius mushroom, jack-o'-lantern mushroom. And there's a little bit more of the defining characteristics about it. This is a common look-alike. Some people will mistake it for a chanterelle, Cantharellus. So, so we're gonna look at that mushroom as well. So it's more or less kind of a vase or funnel-shaped fruiting body. Let's see, differentiated into an upper inner cap and outer hymenium. The gills are more fold-like, kind of that blunt ridge rather than the true gills that the jack-o'-lantern mushroom has. And it's got they've got long, narrow basidia, even though we can't see that with the naked eye. The long, narrow basidia and relatively unspecialized hymenium suggest a kinship between the chanterelles and coral fungi, is what I read in the Mushrooms Demystified. So this is a little bit closer up picture of the hymenium, the golden chanterelle, Cantharellus siberius. It's got blunt, thick ridges, and again, this differentiates it from the jack-o'-lantern mushroom, which is a toxic mushroom. False chanterelles have true gills, although they're forked at the edges. They still appear as close blades rather than lumpy folds. You can see how they differ from the picture that I had before. Okay. Here's some other characteristics that make them different from each other. The jack-o'-lanterns grow in large groups. Chanterelles are usually solitary. Jack-o'-lanterns are more orange, less yellow. Chanterelles will always be near trees, especially oaks and beech trees. And jack-o'-lanterns may appear where there's no trees at all. One of the best ways to identify a chanterelle from a jack-o'-lantern is by examining the gills. Okay. So here's the next one I'm going to go over. It's got true gills that are free from attachment to the stalk. It's got a fragile annulus and distinct vulva at the bottom. This is characteristic of this genus of mushrooms, and it's very common in the eastern United States, but it's also found here in the Midwest as well, mainly in the summer and fall. And this mushroom is actually sometimes confused with puffballs. As you can see, when it's really immature like that, it looks like a little egg, and it's before the universal veil has broken. So unless you cut it open and did kind of a cross section, you might not know that it's an Amanita bisporigia or just destroying angel. So this is another deadly toxic mushroom. Yeah, this is a really cool picture that shows the different stages. I did not take this picture. It's from the Michael Cuyo's site again. So yeah, as the name suggests, it's one of the most poisonous mushrooms out there due to its high concentration of toxins. The resistance of amanitans to all form of cooking and to the digestive tract delay the onset of symptoms to 12 to 36 hours. And also the rapid absorption by cells, especially liver cells, and strong inhibitory affinity for an essential enzyme, RNA polymerase 2, makes it especially toxic and deadly. So good to watch out for that one. This is a common mushroom found in lawns. The cap is oval to convex, becoming plain in age or slightly umbonate. The surface is dry, typically bruising yellow, orange to orange, and then reddish or brown when cut. The gills are broad, free, white, close, and the stalk is white when young and brown upon age. The veil is membranous, white, or with a brown ragged margin. And you can see that there. The spore print's white. And habitat is usually growing in groups or in rings on the ground around trees, especially conifers, bushes, and in the garden near compost piles, ant hills, stables, along roads, and other disturbed areas. And that's the shaggy parasol, also known as Chlorophyllum rhacotis. So here's another one that sometimes you'll find in lawns, and it has a green spore print. So that's a big indicator of what it is. As far as I know, there's from what I've read, there's only one species with green print like this. So watch out for that one. It's the chlorophyllum maldites and green spored parasol. Let's see. And so the spore print is grayish olive to greenish. Habitat solitary to scattered or groups or large rings on lawns and other grassy places, including the garden. Okay. This is a mushroom that's very common here in Nebraska. I see a lot of these. So it's a saprophytic mushroom that grows on hardwood logs and stumps. It's parasitic on living hardwoods. The cap is broadly convex, becoming flat, shallowly depressed or deeply depressed. Pore surface is running down the stem, whitish to creamy, becoming yellowish with old age, not bruising. Pores are large at maturity. The stem is usually off-center or lateral with a velvety dark brown to black tomentum. The flesh is thick, soft when young, but soon becoming corky and tough, especially in the stem. White unchanging when sliced. The odor and taste strongly mealy. Spore print is white. You can see what they call squamules or scales on the upper side. And this is a picture of it when it's mature. You can see how it changes a lot. So yeah, Cerioporus, squamosa, also called pheasant's back or Dryad saddle. Let's see here. That's a mushroom, and it's an edible mushroom for most people. Okay. So this mushroom, it is got smooth or sticky, also called a viscid, yellow-orange to brownish cap and stalk, which is dark brown and velvety at maturity, plus white spores, absence of veil, clustered growth, habit on dead hardwoods. You find these a lot in the winter around Nebraska. And let's see here. Edible but sticky skin that should be removed before cooking. And this is often an important edible in colder regions. Flammulini velutipis, enoki, velvet foot, velvet shank. And this one is a look-alike. It's considered a look-alike with this mushroom. As you start to sensitize your eyes to the different features, you start to think, I don't really think they look alike at all. But that's just kind of the whole process of just observing. And so this one is variable in its color, shape, viscidity, and manner of growth. So it does look different at different stages of the life cycle. There is a veil. It's got a tough, fibrous stalk. It's got these little small dark hairs on the cap. And it's got a bitter taste if you bite into it and spit it out. Growth on hardwoods and yellowish spores. And this is the honey mushroom or Armorillera malea. Variable in its color, shape, viscidity, and manner of growth. I already read that one. This is really cool. It's the Entoloma abortivum or shrimp of the woods. It's just a kind of a ball of tissue with a pinkish area inside, microscopically containing hyphae from Entoloma abortivum and the Amarylarius species. So it has the honey mushroom DNA inside of it. So they found out in 2001, a study found that this mushroom parasitizes the honey mushroom, the Armillarius fruiting, to create this aborted form rather than the other way around, which was previously thought. I think, they probably kind of thought that because honey mushrooms are parasitic on oak trees mainly. So they thought, oh, it's probably parasitizing this mushroom too. But it turns out the mushroom was parasitizing the honey. But I have not found one of those. I would love to. Super cool. Okay, here's another mushroom that is considered an LBM that you'll find around Nebraska as well. Honey mushrooms too. I've found quite a few around here. It has rusty brown spores, unlike the Flammulina and Armillaria that we just talked about. The velvet foot and the honey mushroom do not have brown spores like this one. Small size, thin stem. The veil, which usually forms a thin whitish superior annulus, although the annulus may disappear with age. So here it's kind of difficult to even see it there. It grows on wood. So a good way to tell this deadly mushroom away from the honey mushrooms and enoki is that it has the rusty brown spores. And it's pretty common in the fall and spring. And that is the deadly Galerina autumnalis. Let's see here. And then it actually contains amatoxins like the amanita mushrooms do. So again, a very toxic one. Here's some more advanced terminology. When I was first getting used to the mushroom terminology, I would go through my field guides and just kind of underline or write down words I had no idea what they meant and then just look them up later. But here's just get an idea of all the different words that are out there to describe mushrooms. Here's some major groups of fleshy fungi. I'm going to kind of go over these pretty quickly, but it'll give you just a little bit more idea of the different types. The Agaricus, it's pretty straightforward identification. It's got a stalk, annulus, but lacks a vulva. It's one of the most highly cultivated mushrooms in the United States. The button mushroom and field mushroom are in this family. Lactarius or milk caps. Let's see there. Yeah, I won't read through all of this. Cantharellus or Chanterelles. Boletes. The amanitas. Rhodes. The oyster mushrooms are in this group. Tooth fungi, Hericium, Dentium. These are really good for eating. The lion's mane mushroom is really delicious. And I actually found a lion's mane here in Nebraska one time, which was really exciting. Let's see. Puffballs and earth stars, Lycoperdales. Here's some more parasol, the Lepiota. Some Latin name suffixes and prefixes. These are just really helpful to know because you see them in a lot of different mushroom names. Cybele means cap, phylum means gills or leaf. Apo means open. Guys yourself with some of these prefixes and suffixes can just help understand the mushroom a little bit more, at least help remember the Latin name. And yeah, just lastly, a little spiel on mushroom cultivation and how you can ethically do that. There's a lot of debate about what's ethical when mushroom hunting, but they talk about either plucking or cutting the stem or twisting it completely from the ground, uprooting the mushroom, mentioning how the mycelium can be damaged, impairing the future growth. And the truth is it kind of depends on the mushroom and also the substrate that it's growing in. So saprophytic mushroom may come up with a lot of mycelium, especially if it's kind of feeding on leaf litter, loose material like that. But like a mycorrhizal mushroom tend to detach pretty cleanly from its parent mycelia. And basically just the main risk is habit destruction. So as long as you're kind of paying attention to what you're damaging as you're pulling that mushroom, or if you're damaging it, just be really careful, really mindful of the environment that you're in. And polypores, perennial mushrooms will obviously take a lot longer to grow back. And then lastly, there's just this little graph that I found here that can kind of help you gauge the mushroom abundance in the area that you're hunting in. So, taking into account the different resources that are available to that mushroom that year, that season, will help you kind of understand how to ethically harvest it. So that's about the end of my presentation. If you guys have any questions for me, I know that was just a ton of information, but I like to kind of get the foundation and the basis of using field guides. There's just a lot to familiarize yourself with, but it's a process, and I think it's a really fun hobby to have. I really love it. So thanks, guys.